Hi there, and welcome to this video on GCSE Biology for the AQA Specification, focusing on the topic of animal tissues, organs and organ systems, and in particular the human digestive system. I'm Shumina from StudyMind, where we help you revise GCSE Biology with our helpful video tutorials, tailored to your subject, your specification, and to you. If you're new here, make sure you click the subscribe button. Whilst you are watching, please leave any comments below if you're unsure about anything, and let us know if it's your first time watching our videos so we can send you our free revision materials. We also have helpful timestamps below for each part of the video to help guide you through the specification. So let's get started. Hello all, and welcome to tutorial 1 of 7, in which we're going to be looking at the human digestive system. So, in today's session, we are going to be looking at the following components. So it's going to be quite a lengthy tutorial, so do feel free to pause anywhere and go back and replay it, because it's quite important that you understand each part before we move on to the next section. So, remember from last session, we had a look into the definition of an organ system. Well, the digestive system is a really good example of an organ system, as several organs work together in order to digest and absorb food. So several organs work together in order to reach one function. So, the digestive system is involved in the digestion and absorption of food. So, what is digestion? Well, it's the breakdown of food from large, insoluble molecules into smaller, more soluble molecules. So this means that not only are the food particles broken down into physically smaller components, but also they can be more easily absorbed. And so what does absorption actually mean? Well, it involves the small soluble molecules that have been broken down in digestion being taken up into the bloodstream, where it can then be utilised by the body. So these are all really key definitions, so you're going to have to memorise these, and these are really easy 1 markers that you can get in your exam. So let's take a look into how the process of digestion actually works. Well, as you can see here, large insoluble molecules, such as these, cannot be absorbed into the bloodstream. So there's multiple layers that it has to go through in order to go from the lumen of the ileum into the bloodstream. And just bear in mind that the ileum is the final section of the small intestine. And so we want to absorb these food particles into our bloodstream, because only then can our body actually utilise the nutritional content in our food that we eat. So the problem that we face here is that large insoluble molecules that have been broken down by your teeth, for example, and enzymes in your saliva, which we'll come on to look at a little later, aren't sufficient enough to allow absorption to the bloodstream. We're going to need even further breaking down in order to get these smaller, more soluble molecules of food. And this is the large insoluble molecule. So, this is where digestive enzymes come in. So, digestive enzymes hydrolyse these large insoluble food molecules into smaller, more soluble molecules that can then be absorbed from the ileum across the epithelial cell lining and into the bloodstream. So, just to put it simply, the food is broken down into smaller pieces which can then be transported across cell membranes in the small intestine through to the bloodstream. So overall, digestion involves breaking down large molecules of food which are too big to be absorbed into smaller, more soluble molecules of food which can then pass easily into the bloodstream. And this is really, really vital. So this is the key function of the digestive system: the breakdown of food and the absorption of nutrients. So, as we said, large insoluble molecules cannot be absorbed into the bloodstream, and hence they must be broken down into smaller, more soluble molecules which can cross cell membranes in the small intestine. So let's look at the types of digestion that you can get. Well, there are two. First of all, there's mechanical digestion, which is the physical breakdown of food. So here, for example, this can occur in your body by your teeth chomping down on food, breaking it up into smaller particles, and also by your stomach, because stomach contractions break up food particles too. So as the stomach contracts in and out, it will also break up food particles into smaller, physically smaller pieces. But there is also enzyme-driven breakdown via chemical digestion. And this is because mechanical digestion alone is not enough. So this chemical digestion occurs by multiple enzymes, but in this tutorial and for your exam, you just have to know about lipase enzymes, protease enzymes, and carbohydrates enzymes. And we're going to be looking at this in a lot more detail. So first of all, we have our physical breakdown of food via mechanical digestion. So as your food passes through your mouth, you break it down via mechanical digestion and then down your esophagus into your stomach, and we also get the churning process in your stomach, which breaks it down even further into smaller pieces. But there is also enzyme-driven breakdown via chemical digestion. So just bear in mind that it's kind of like a layering process, and each stage of this digestion is really important, because the mechanical digestion, although it's not sufficient to allow the food particles to be absorbed, it's the main point of it is to break the food particles into smaller pieces so that they have a larger surface area for the enzymes to then act upon them and allow for chemical digestion to take place. So even though mechanical digestion alone is not enough, it is crucial in order to facilitate chemical digestion to then occur further downstream. So now we're going to come on to a diagram of the digestive system, and this is just a simple example of a kind of learning process where you just have to keep testing yourself. So perhaps print off this diagram and cover up the labels with like post-its or something, and just keep testing yourself now and again, because these are really, really easy 6 or 7 markers that can come up in your exam. Really easy marks if you have looked at the diagram at least 3 or 4 times before your exam. So we'll start off with an easy one. That's the mouth. So that's essentially where your digestive system starts in the whole food processing timeline. We then come on to the esophagus, which is the pipe that leads from the mouth into the stomach. So food is transported down from the mouth into the stomach via the esophagus. Then we come to the stomach. There's the liver, which forms part of the digestive system. The small intestine. The large intestine. The rectum. And finally the anus. So we're kind of trying to go from top to bottom. So I really, really highly suggest that you print out this diagram and continually test yourself. Maybe even try to draw it out. And we're going to have a look at the functions of these organs too. So the mouth is really, really crucial in mechanical digestion, as we saw earlier. So our teeth chew up our food, and this breaks down large food molecules into smaller food molecules. So here we can see big proteins being broken down into smaller proteins. And also, we also have chemical digestion taking place in here. So remember, chemical digestion is the breaking down of molecules using enzymes. So first of all, we have the teeth chewing up the food, which is our mechanical digestion. And remember that mechanical digestion breaks up our food molecules into smaller pieces, which the enzymes can then act on in chemical digestion. And the key enzyme I want you to remember in the mouth is amylase, which is produced by the salivary glands. And amylase is really important in digesting carbohydrates into their smaller components, so maltose, fructose and glucose. But if this is confusing you slightly right now, don't worry, we're going to come on and look at that in a little more detail. Now we move on to the esophagus. So we've had the mouth, we've had chemical and mechanical digestion. So now these smaller molecules of food are going to pass down into the stomach. But in order to get from the mouth to the stomach, they've got to pass down the esophagus, which is this section here. So it links the mouth to the stomach. And food doesn't just drop down this pipe. What happens is the esophagus undergoes sequential contractions because it has a muscular wall. And this is going to move your bolus of food, or ball of food. Bolus is just another word for a ball of something. And so these muscle contractions are going to occur sequentially down this pipe, which is the esophagus, and that's going to cause an overall movement of the food particle down. And actually this muscle contraction is called peristalsis, but we don't need to worry about that word so much right now, but you may have heard of it before. Then we come to the stomach. And in the stomach, protease enzymes, pepsins, are released to digest proteins. But also, the stomach has this very, very low pH of pH 2, which means it's an extremely acidic environment. And this is going to kill any bacteria present in the food. So not only is the stomach involved in digestion in the sense that it further breaks down food particles in mostly proteins, but also it has a protective mechanism, which is the acid killing of bacteria. Now let's have a think about the pancreas. Well, the pancreas is extremely important in the production of enzymes, and it produces digestive juices which contain all these enzymes, and these enzymes are really critical in the chemical digestion of proteins, which are the proteases, lipids, which are broken down by lipases, and carbohydrates, which are broken down by carbohydrases. The liver produces bile, and bile emulsifies lipids and breaks lipids. Lipids is just another term for fat, into smaller fatty droplets. So bile emulsifies the lipids, breaking them into smaller fatty droplets and aiding their absorption. And we'll look at that in a bit more detail in a while. The small intestine is really, really important in absorption. So it has these structures called villi, which basically, if this is your surface of your small intestine, they would, if you zoomed in on this section, try it up here, it would appear like that, and those are your villi. And villi just increase your surface area for absorption, but we'll be looking at this in a later tutorial. So the small intestine has many of these villi which allow for efficient absorption of nutrients that have been digested. And these villi, they have a thin wall which allows for a short diffusion distance, which means the food particles only have to travel across 1 or 2 cells in order to reach the blood. Villi also have a rich blood supply which maintains the concentration gradient for nutrient absorption. And as I said earlier, villi provide a very large surface area to aid the speed of diffusion and active transport. And these enzymes that are shown on this slide, proteases, carbohydrates and lipases, we're going to look at in a little more detail in a minute. And as I said, the small intestine is really crucial in absorption of digested products. The large intestine, on the other hand, is really crucial in the absorption of water and ions. And actually, this is how faeces is produced from undigested food, because the water is absorbed out of the undigested food into the bloodstream, and the undigested food is left in the large intestine, where it eventually compacts together to form faeces. And any undigested food is then stored in the rectum, where it is then voluntarily expelled through the anus. So this is all summarised in this table. So I'd really suggest you write this up yourself and highlight the key terms. So for example, in your mouth, you're going to have to remember that you have mechanical digestion and you also have chemical digestion. So I think that's a really good way of remembering the key functions of each part of the digestive system. So now let's take a look into the varying food groups. So we're going to divide them up into carbohydrates, proteins and lipids. So carbohydrates and lipids are made up of carbon, oxygen and hydrogen. Proteins are also made up of carbon, oxygen and hydrogen, but they also have nitrogen in them. So let's have a look at the digestion of carbohydrates. Well, let's look at the largest form of a carbohydrate, which is a starch molecule, which is many, many individual units joined together. And this is broken down by carbohydrase enzymes into maltose, which can then be broken down even further into glucose, fructose and galactose. So note how we go from long chain up here to small chain. And this is basically your process of digestion. This is a common theme that will occur throughout the digestion of carbohydrates, lipids and proteins. So it's these carbohydrase enzymes that are breaking down your carbohydrates. But this occurs in two stages, as I described on the previous slide. We get starch to maltose breakdown, and then maltose to smaller, more simple sugars such as glucose, fructose and galactose. And carbohydrases are found in the mouth, for example, such as amylase, which hydrolyzes starch. But also the pancreas produces amylase that travels to the duodenum, which is a part of the small intestine, via the pancreatic duct, and this allows for further digestion of starch. So starch is broken down into maltose in the mouth by amylase, but it can be further broken down into lots of these smaller, more simple sugars such as glucose, fructose and galactose in the small intestine. So similarly, protein can be broken down sequentially. And if I just go back here. So protein is broken down into dipeptides, which means two kind of small molecules of the basic subunit of a protein, i.e. amino acids, by endo and exopeptidases. So endo or exopeptidases. But then dipeptides are broken down into amino acids by dipeptidases. So we have the exoendopeptidases here, working on protein being broken down into dipeptides, and then dipeptidases breaking the dipeptides into amino acids. And note the common theme, it's this ase, which shows that it's an enzyme that's breaking down the dipeptide into amino acids. So proteins, like carbohydrates, are also broken down in two stages. First of all, the protein is broken down into a dipeptide, which contains two basic subunits, and the basic subunit in a protein is the amino acid, and the dipeptide is then split into individual amino acids. So where are proteases found? Well, the protease enzyme pepsin is found in the stomach, and it converts proteins into amino acids. And the optimum pH of pepsin action is 2, which is the pH of the stomach. So this means that pepsin is in a really optimal environment for its action. But also, the pancreatic juice that is released into the small intestine contains protease enzymes, including trypsin, and this converts proteins into amino acids, like pepsin. And finally, let's look into the digestion of lipids. Well, lipids are formed from one glycerol molecule and 3 fatty acids, and they are broken down by lipases. So again, note the common theme. That's your lipid, and it's being broken down, so we're going to have this A's stem on the end. So it's really easy to kind of remember which enzymes are responsible for breaking down the larger molecule. If it's a lipid, it's a lipase. If it's a carbohydrate, it's a carbohydrase. If it's a protein, a little bit different here, a peptidase. They all have this A's stem. So lipids are broken down into their smaller components, fatty acids and glycerol, by lipase enzymes. And lipases are made in the pancreas and secreted into the small intestine via the pancreatic duct. And it's really crucial for you to remember the structure of a lipid, which is 3 fatty acids and 1 glycerol joined together. And let's have a look at where lipids are found, or lipases are found. So pancreatic juice contains your lipase enzymes, which are secreted into the small intestine. And just bear in mind here that lipids and fats are used interchangeably, but do try and use the term lipid because it does sound a lot more scientific. So just to summarise, we have our proteins being broken down by proteases into amino acids, and this occurs primarily in the stomach, where the optimum pH for pepsin action is pH 2, and therefore that's a really good environment for protein digestion to take place. We have our carbohydrates, which are being broken down in our mouth and our stomach via amylase. And then we have our lipids, which are being broken down mainly in the small intestine via lipases. So earlier on, I briefly mentioned about bile. And we didn't really look at this in a lot more detail other than saying that bile is responsible for the emulsification of lipids. But what does that actually mean? Well, before lipids can be hydrolyzed by lipases, they need to be emulsified. So emulsification is the process by which large lipids are broken into tiny lipid droplets. Let's just go back to the last slide. So that's really important for you to remember, that emulsification is the process by which large lipids are broken down into tiny lipid droplets, and this is a really easy 2-marker that AQA can ask you. So these 2 points here are really, really important for you to remember. But the whole reason that this is really, really crucial is that this breaking down of the large lipids into tiny lipid droplets increases the surface area of lipids in order to speed up the activity of lipase enzymes. So as I mentioned earlier, bile is a substance produced by the liver and it's stored in the gallbladder. But during digestion, bile is released from the gallbladder and transported to the duodenum via bile ducts. And this is where, in the duodenum, that bile then emulsifies the lipids. And just note that bile is alkaline, whereas the stomach is an acidic environment, remember, and it has a pH of 2. So pH 2 here, and since bile is alkaline, it must have pH above 7. So this is going to neutralise the food in the stomach acid. So this is actually really, really handy because I said that proteases work well in the acidic environment of the stomach, but trypsin, lipase and carbohydrates have to act at their optimum pH, which is at a more neutral pH. So yes, pepsin works well in the pH 2 of the stomach, but we also need to allow the other digestive enzymes to work well. And in order for that to occur, when the food moves out of the stomach into the small intestine, it is neutralised by the bile so that then trypsin, lipase and carbohydrates can act at their optimum pH. Now, we've had digestion occur, so we've absorbed these smaller molecules up into our bloodstream, but what happens from there? Well, once the products of digestion are absorbed into the bloodstream, the body can use it to build molecules. So, for example, the simple sugars that are absorbed, such as maltose, fructose and glucose, can be used to build new carbohydrates. So, glucose can be stored as glycogen in the liver, and this provides an energy store. Similarly, amino acids absorbed can be used for building protein. So bodybuilders often drink lots of protein shakes and eat lots of chicken because these all have high contents of protein and therefore allow the increased number of amino acids absorbed by the body to be used for muscle synthesis. And likewise, lipid-wise, the fatty acid and glycerol molecules absorbed from the small intestine can be used to store fat. So if you eat lots of fatty foods, you are likely to build up more and more fat stores, again for energy. So glucose can actually be used to release energy via respiration. Because remember, the respiration equation is glucose plus oxygen goes to carbohydrate plus water plus energy in the form of ATP. And this is why food is so important for daily function and especially for exercise, because glucose allows for the production of ATP, which can then be used for muscle contraction, for example. Now let's take a further look into how enzymes work. So, if we were to label this diagram up, we've got our enzyme, and it has these crucial parts in it which have a very specific structure called its active sites. And this is where our substrates fit in. And you can see that they have very specific complementary, which is a key word here, sites on their own structure that fit into the active site of the enzyme. And this is how an enzyme-substrate reaction occurs. So enzymes are biological catalysts, which means that they increase the rate of the reaction without having to increase temperature. So they do this by decreasing the activation energy required for a reaction to occur. Because when two reactants collide, they need enough energy for a reaction to happen, and this is known as your activation energy. But enzymes overcome this threshold that needs to be reached by reducing the activation energy, and therefore this increases the rate at which the reaction occurs. And it also allows for reactions to occur at lower temperatures and at a greater rate. So how does this work? Well, the active site of an enzyme binds to a substrate, and the structure of the enzyme, of the active site of the enzyme, determines which particular substrates it is capable of binding to. And because the active site has such a unique structure, it means that most enzymes can only bind to two substrates which fit specifically into its active site. So therefore, substrates must be complementary to the active site. So let's just take a look at this in a little more detail. So enzymes are proteins, which means that they're made up of sequential structures. So the primary structure of a protein is the sequence of amino acids. So it's just the 2D sequence of amino acids. This then builds up to the secondary structure of a protein, which is the way in which it folds. So it can fold to form an alpha helix or a beta pleated sheet. The tertiary structure involves more bonds forming between the pleated sheets or the alpha helices, and therefore allows their interaction. And the quaternary structure is the interaction of multiple tertiary structures. So this means that the protein has a very, very specific structure overall. And this is why the structure of the enzyme is essential for its function, because it's a protein, after all, and it has this very specific structure. So as I said, the order and number of amino acids in the chain forms the primary structure, which is flat and 2D. The secondary structure is how the protein folds or coils to make a 3D structure. So this can form an alpha helix or a beta pleated sheet. And how does it do this? Well, hydrogen bonds form between amino acids. The tertiary structure, on the other hand, is the final 3D structure of the protein chain. And it doesn't only involve hydrogen bonds, but also many other bonds, such as ionic, disulfide, hydrophobic bonds. And this determines the shape of the active site, and it's very easily affected by the environment, so the temperature and pH. And finally, the quaternary structure is how different chains in the protein come together. And this forms the final structure of the protein. Now, don't worry too much about the bonds at each level, but do know the definition of what each level means and what is involved. So, because of this specific structure of proteins, enzymes tend to have optimum conditions. So, enzyme tertiary structures are extremely sensitive to their external environment, as I said earlier. And changes can cause a protein to denature, which means that it can no longer bind to its substrate as effectively. Because remember, the enzyme has a very, very specific active site structure. So environmental changes can cause a change to this active site, which then means that the substrate can no longer bind to its specific binding site, the active site on the enzyme. So, for example, let's look at temperature. So, as we increase the temperature, we're going to get an increase in the rate of reaction because the molecules are colliding faster and with more energy. So there are more collisions overall and more of them are successful. Then we reach our optimal temperature, which is roughly at body temperature for many enzymes in the human body, which makes sense because we want our body enzymes to function really well at the natural temperature of our body. And this is where the rate of enzyme activity is highest. But after that, we get this denaturation, and this is because the higher temperature breaks bonds such as hydrogen bonds in the tertiary structure of the protein and therefore causes a change in the active site. This means that the substrate can no longer bind in the active site, and therefore we get a slower rate of reaction. So, as I said, the initial increase in the rate of enzyme activity as temperature increases is due to more frequent collisions, because the reactant particles are moving faster and they collide more often, but also more successful collisions. So any collision is more likely to be successful because it has more energy, because of this higher temperature. But then eventually, as the temperature surpasses around 37 degrees, we're going to start to break our enzymes' bonds, which changes the shape of the active site. The rate of reaction then drops rapidly, which means that the rate of enzyme activity drops. So very low temperatures or very high temperatures are not ideal for successful reactions. And similarly with pH, we're going to have our optimum pH that the enzyme likes to work at, and that's going to be a neutral pH of our body, which is around 7.4. But of course there are exceptions. So remember earlier we said that the protein pepsin, which works in the stomach, works best at a pH of 2 because that's where it functions in the acidic environment of the stomach. So as we start to get any kind of variation from this neutral pH of 8 in most enzymes, we start to get a disruption of the bonds in the tertiary structure. And this leads to an alteration in the specific shape of the active site, and therefore the substrate may no longer be complementary to the active site. So make sure you learn these graphs and understand what is going on, and it might be really useful to draw them out and annotate onto them what is happening at the different stages around the optimal temperature or around the optimal pH, because it's a really, really common point of examination by the AQA examiners. So, just to summarise, we have our enzyme, our specific shaped active site, which is complementary to a single substrate. So this actually is known as the lock and key model. And so when you insert a key into a lock, the key has to have a specific shape in order to unlock the door, for example. So it works the same here. The substrate has to have a specific shape. The substrate is the key, the enzyme is the lock. And the substrate must have this correct shape which allows it to fit into the active site of the lock, which is the enzyme. But there are some problems with this model, because it assumes that the enzyme active site and substrate have the exact same shape, and no change occurs when they bind. But this isn't quite true, because enzymes can bind to many different substrates, although specific substrates I do have to say. So in order to do this, they may actually change their active site slightly in order to bind to a new substrate, which is where the induced fit model comes in, which is very similar to the lock and key model, but it just states that there might be a slight change in shape of the active site in order to accommodate the substrate. So the lock and key model assumes that the enzyme active site and substrate have exact complementary shapes, but the induced fit says that perhaps there is a slight conformational change that occurs. And lastly, let's do a little bit of maths. So I know that sounds a bit daunting, but it's all very, very simple. So in your exam, they might ask you to calculate the rate of a reaction. So let's just think about it. In order to look at whether a reaction is occurring or not, we can look at how quickly a reactant is used up, or how quickly a product is formed. So therefore, we can derive these two equations. First of all, that the rate of reaction is equal to the change in amount of reactant over time. So usually what will happen here is the amount of reactant will decrease as it forms the product. Or equivalently, we can look at the change in amount of product, which will increase over time. So these two equations are ones that you should learn. And often you may need to calculate the rate of reaction from a graph. And all this involves is finding gradients. So we'll work through this step by step. So first of all, you have to check the graph's axes. So most graphs like this one will show you a product time graph, with the product formation on the y-axis and the time of reaction on the x-axis. And you can see that the time of reaction is measured in minutes and the product formation is measured in grams. You then need to pick 2 points on the graph, and you need to remember to pay careful attention to your units, okay? And so you pick 2 points on your graph and you find the gradient, which is just the rise of the slope. So this is your rise over your run. So that's your simple gradient equation here. And you're just going to divide your rise by your run. So here that would be 10.4 grams over 2 minutes, which is 5.2. Now remember your units here because, whoopsie, got a bit excited. Remember your units because we've said that the gradient is going to be 10.4 grams over 2 minutes, which we're saying is 5.2. Now a really simple way of working out what the units of your answer is to simply just treat your units here the same as you treat your numbers. So it's going to be 5.2 grams over minute. But the more colloquial scientific way of writing this is 5.2 grams per minute. Do you see how we've done that? So we've literally just said it's grams formed per minute, and that's your units here. So then do check your gradient. So check that the gradient is constant throughout if your graph is a curve. And in this graph, we could say that the rate does decrease as the reaction continues. So just be aware of what is actually going on in your graph overall. So that's all for today. We really have covered a lot, so well done for getting through it. And I shall see you for our next lesson. Thanks for watching this free video from StudyMind. If you liked this video, make sure to subscribe to catch our newest videos by clicking below, and leave a comment on a topic you'd like a video on. Click here to watch more videos in our series for GCSE Biology, or visit our website studymind.co.uk for free past paper compilations by topic and specification.